Mendelian genetics. Gregor John Mendel was one of the first one who succeeded in discovery of the basic principle of genetics. We can say genetics is the first scientist who did experiments on genetics and also succeed in his experiments. He did his whole experiments on Pisum sativum or pea plant, 1856, about for 88 years. Later, Mendel presented his research finding to a magazine called Natural History in 1865. Then, after 1 year, they republished it, but none of the scholars of that time give attention to the Mendel. So, for 34 years, nobody gave attention to the Mendel studies because the scholar of the time were preoccupied with the theory of Darwin, means their mind was busy with organic evolution. Then the three scientists, the various Correns and the Chermak, the three scientists were interested in studying and function and behavior of chromosomes, studied on Mendel genetics and the hypothesis, and as we remember from the chapter 1, it became the golden age for cell biology after these three scientists. We said Mendel is the scientist who did experiment and succeed. One reason of this success in experiments about the best model he chose in his experiment. So all organisms cannot be used directly in genetical experiments. There are about 6 properties organism must have to be used in genetical experiments and also succeed in these experiments. Short life cycle, large amount of production to see a lot of probabilities, having the possibility to get variations or genetical mutation when organism exposed to unsuitable environmental like radiation and chemicals. And to see these variations, said need to produce large number of production in each generation. Ability to control fertilization. So if it is a plant, must not do self-pollination, so we choose the model of plants which avoid of self-fertilization. Easy upbringing and maintenance, make it easy and cheap. Ability to produce new structures as a result of sexual reproduction, so an organism which can reproduce only by asexual reproduction, we cannot see different variations because all organisms will be same, or maybe it must be affected by delivery of transmission of genetic material from one organism to another, like by viruses. Many research conducted on bacteria, like research are conducted on this kind of organisms. Also, most of the genetic problems are focused on for Mendelian genetic, like Pisum sativum pea plant, or corn, or with barley, squash, snapdragon flower. In some animals like Drosophila, mice, chicken, pig, cattle, and also human. So the question which we're going to study on about the genetics are focused on these organisms. Usage of genetic models by Mendel in experiments. We said that Mendel is the first scientist who did experiment and also succeed in experiments because there are reasons. Was one of them because he was choose the best model for experiments, because the best model, which is pea plant, short life cycle, easy upbringing, a lot of production, avoid self-pollination, and also affected by what? By mutation. So Mendel succeeded in experiments because he chose the best model. In second point, Mendel limited his research on one pair of traits. One pair of traits means he did one set or 100 of experiments focused only on one trait, like color of flower. Another set of experiment only about position of flower. Another group of experiment focuses on only length of plant. It is short or tall, so he succeeded in the experiment. And the number 3, the Mendel keeping accurate records in his experiments Like what? Like in this picture, for example, when he did experiment between two yellow and green plants, he recorded the numbers like 6022 or 2001, or the ratio 3.01 to 1. So this accurate records caused Mendel succeed in his experiment. In addition, this table which we see about the character or genetical trait for pea plant, which is used by or in experiments of Mendel, mainly the question about this plant, also about this traits. The flower color, flower position in axial or terminal. Axial is on lateral position, flower terminal located on the top. Seed color, yellow or green. Seed shape, round or ringlet. Pod shape, inflated or constricted. Pod color, green or yellow, and stem length can be tall or dwarf, or we call it short. Some genetics terminologies and symbols which we need to know or understand this terminology or terms to solve the monohybrid or dihybrid genetical questions. So from these four terms that we see, we may familiar with gene, because we heard the name of gene in chapter 1 when we study on nucleus. In nucleus, if we recall the information about the nucleus covered by nuclear membrane, filled with nucleoplasm, contain what? Chromatin network and nucleolus. This chromatin network with the filamentous structure of DNA, but during cell division, it shortened it and thickened it to form basilar structures known as chromosome. I would say the chromosomes are carry genes. So the gene is a sequence of DNA, a part of DNA on chromatin network or on chromosome. This has a specific function, like what? Turning the genetic code to a protein. Like this gene contains information to form a protein, we call it hemoglobin. Or controlling expression of character, like a piece, a small part of DNA, a sequence of DNA is involved for what? For flower, color of flower. Also, it's another gene. So a gene is a part of DNA in chromatin network, or a part of DNA on single locus of chromosome, which turn the genetic code into a material protein or control a character and prove itself through the contrast alleles. Contrast alleles means what? Like if the flower color for pea plant, we said purple and white. When it is not white, it is purple. Or in absence of purple, the color is white. So these two opposite traits in Mendelian genetics, we call them contrast alleles or contrast genes. Then allele is alternative form of a gene. As we remember from the meiosis, we said there's the chromosomes makes couple, this two or couple of chromosomes, which one received from father, one received from mother, and they have similar function. We call it them homologous chromosomes. So homologous chromosome, they contain similar genes. How there's a gene about flower color on this chromosome, on its homolog also, there's a gene about flower color. And when these two chromosomes found in the same organism at the same time, we call this organism is diploid. So these two genes which have similar function, we call them allele genes. As we said, the contrast alleles or contrast traits in Mendel, the flower color can be purple color or white color. So like this one for white and that one for purple. So the color of flower can be white or purple, but there is no a third chance for color in flower. So these two genes which have similar function, but on appearance of organism they show different effects. We call them allele genes. Gene expression is the process of using DNA information by cells in the manufacture of a particular protein. As we said, this part of gene used to make a protein. So the process of conversion this genetic information into a material or a protein is gene expression. Genotypes reflects the composition or structure of genetic of individual. Means all genetic material in an individual is genotypes. It expresses genetic codes together in a particular individual, like homologous chromosome or all genes found in nucleus together in one organism. So we in problems we show or symbolize the genotypes with letters with A or with B or with T. So that form of writing it shows the genotype of organism. Phenotype or phenotypic category, it refers to the properties or observed sign of organism which are genetically controlled. If we do simply a chromosome, on this chromosome, capital T, like show the tallness of organism, for example. So this T is a gene. So in another chromosome, also there is a gene, also it is capital T, also it's tall. So these two genes, they are allele genes. And these genes, when we show them by letter, it's genotype. But when we see a plant with tall stem, it's phenotype. So the observed sign of organism which are genetically controlled, we call them phenotypes. For example, the stem length of stem shortness in PSM cytopenia refer to the expression of natural information. These are available in the genetic factors. By following the principle of dominant and recessive traits, we can tell about the phenotype for the individual from the genotype. But what does the dominant and the recessive traits mean? So if an individual which is diploid contain T capital allele genes, so the stem length will be tall. Another organism, T capital with T small, the organism again will be tall stemmed P plant. So if the genotype is not look like this, T capital means pure, but one capital, one small, like two opposite traits, tall and short. But which trait is appear on organism, this trait is dominant, but which is not appear, it's recessive. So with T capital and T small mean tall, one gene tall, one of them short. But if organism is tall stemmed, the tallness we call it dominant, but the short trait we call it recessive trait. For example, T capital, T small refer to the long stem, but T small and T small refer to the short. So an organism, if T small, the next chromosome also T small, so plant will be short stemmed P plant. Why? Because there is no dominant, two recessive. They show their effect on appearance of organism. When the individual carries genetic factors like symmetric, like T capital or T small, we call this homozygous or homogeneous or pure. But when individual carries genetic model which carries not symmetric, like T capital, T small, we call this organism heterogeneous or heterozygous, but mainly we use the term hybrid. Before passing the solving the questions, we need to know some symbols in addition to the terms which use it in genetical question or problems. F1 is first generation, but this F not about the first, is about the filial, and that it means offspring. First generation F1. G, it comes from the gametes reproductive cell, which produced by major organisms by meiosis. P, parents, is X or cross or marriage or mating or vaccination or hybridizations, or sometimes we use marriage between crossing two organisms. The male sign and female sign also we use in genetical experiments. Let's do a simple example to learn about the terms and the mechanism of solving genetical question in Mendelian genetics. A tall stemmed, pure pea plant crossed with another white or short stem pea plant. Find the genotype and phenotype for F1 and F2. So because it pure for both traits, so we write a tall stem pea plant with another short stem pea plant, male and female. Pure, capital T we use for tall, small and small for short stem pea plant. They are our first parents. This writing is phenotype by symbolizing with letters is genotype of what? Parents. So the parents produce gametes through meiosis. So meiosis, two homologous chromosomes or these two genes are separated one from another, each received for one by one gamete. So number of chromosomes reduced to the half. Capital small t also produce one gamete. There's a simple and important rule. Always, if the genotypes are pure, they produce one kind of gamete. Pure dominant produce one kind of gamete. Also pure recessive produce one kind of gamete. And F1 generation, we know new organism formed by what? Union between male and female reproductive cell. So F1 generation, genotype T capital T small, phenotype hybrid, tall, stemmed, pea plant. Ratio 100%. To find F2 generation, we take organism of F1 generation and leave it for self-pollination. So tall stem pea plant with another, again tall stem pea plant. Second parents, T capital T small and T capital T small. They produce gametes by meiosis again, but at that time they are hybrid but not pure. So hybrid traits, again a simple rule, produce two types of gamete, one capital and one small. one capital and one small. New generation, means F2 generation, formed by union between male and female reproductive cell. One male, one female, capital T, capital T. One male, one female, capital T, small t. One male, one female, capital and small, and one male, one female, small t, small t. They are genotypes of second generation. The phenotypes is 3 tall stemmed pea plant, while one of them short stem. The phenotypic ratio is 3/4. Why? Because from 4 organisms, 3 of them tall. From 4 organisms, 1 of them short stemmed pea